Thank you so much. It's really an honor to be here and standing up here. I guess I've done pretty well if I've managed to not have to give rounds until 6 years after my completion of my fellowship, but I really am delighted to be here today. So although I was born in Limerick, I am not going to give my seminar in the form of a limerick. I apologize. It'll be a regular seminar. So I only have one disclosure that's pertinent to this talk, and that is honorary and research funding from BMRU. The rest of my disclosures are in the pieces of paper that many of you have. So depending on your age, the better, faster, stronger, you might be thinking of the $1 Million Man or Daft Punk, or if we've got some really wee ones in the audience, this gentleman here. And I put this up because I think that that is a very pertinent quote that we should all be telling ourselves every day, to believe in your finest and conquer your shyness. So these are my learning objectives. They're listed here. And I'm going to begin by giving you an overview of a timeline of diagnostic microbiology. So the point here is not to be all-inclusive with respect to major discoveries in clinical microbiology or in diagnostics within the field of clinical microbiology, but mainly to just show you some major discoveries, and also really to point out that despite the fact that bacterial culture and gram stains and other stains were first invented in the 1800s, we're still using these techniques today, and I'll hope to convince you that that's not a bad thing. Just to put into context of what I'm talking about here today, in 2006 is when the first MALDI microbial identification system was launched. Okay, so obviously knowing that we have PCR, and I will say my Irish accent makes me say or instead of R, so I apologize, so PCR. Why can't we just do PCR for everything? Well, there's a couple of reasons that we don't really want to do PCR for everything, and that is because just something as simple as a gram stain can be very advantageous. It's very fast. For example, if a patient is suspected of having bacterial meningitis, we typically will turn those gram stain results around within 30 minutes or so. It can also provide very important information on the quality of the specimen. And importantly, a presumptive diagnosis can often be made if organisms are observed. Why do we want to continue to do bacterial culture and culture of other pathogens? Bottom line is that it's more sensitive than microscopy. It's relatively cheap compared to molecular methods. And this is really key, that you have to have the organism growing in culture to be able to perform susceptibility testing. So you might say, well, look, we could just do PCR, find the target, and be done. But the problem is that the bugs are clever, and they're continuing to evolve new resistance mechanisms. So an example of this is a paper that was published in 2011, where they basically discovered this new resistance marker that was associated with methicillin resistance in Staph aureus called MEC-C. Now, regular Staph aureus that we're used to has a MEC-A gene. And so if you were only looking for the presence of the gene, you would have missed the fact that these isolates were methicillin resistant and that beta-lactam therapy, with the exception of ceftaroline, would have failed for patients with these infections. Okay, so that's the reasons we want to continue to do culture and gram stain. But just to put it into context, what are some of the disadvantages of doing culture? Okay, so this is a pretty typical scenario. We get the specimen in the lab, we do a gram stain, and we plate it to a mixture of solid and liquid media. And we, on the first day, observe within a couple hours and report it, there are 3 plus polys present, as well as gram-positive cocci clusters. So the next day, because the bacteria have to grow up and to be able to work with them, we see that there's growth on the plate, and we can do some spot tests and report it as Staph aureus. Because of Staph aureus, we can also do some spot tests to look for PBP2A, which is the protein that's encoded by MEC-A that confers methicillin resistance. But the full susceptibility results are not going to be available until a day later. Now that's kind of the best case scenario for a bug like Staph aureus, where we can use spot tests to identify it. But the problem is that for many, if not most, gram-negative frauds, you're now delaying your identification by 18 to 24 hours using standard identification methods. And then if it's a mixed, if there's mixed colony morphologies separated, those all those all have to be separated out in order to have enough growth to be able to work with to do identification and susceptibility testing. So bottom line is that unfortunately this is how we're rocking it in microbiology land, and I'll get back to why this is significant later. Okay, so I'm not here to tell you about traditional culture. I'm here to tell you about MALDI-TOF. So for all you mass spec aficionados in the audience, I'm going to be keeping this pretty basic. So I apologize to all the folks that are really down with the mass spec. I'm going to keep this simple. So as we apply this to microbiology, what we're dealing with is a target plate to which the colony is directly applied. There are different steel plates versus plastic disposable plates depending on which system that you use. These are then covered with a matrix that imparts a positive charge to the proteins that are present, and then these proteins then fly through a field-free vacuum where they reach a detector. And so the time of flight refers to the time they're traveling through this tube. And what results is the production of a characteristic spectrum that's unique to that organism. And then this can then be queried against a database to result in an identification. So since the introduction of these commercially available systems for identification, you can see that there just have been an ever-increasing number of publications that have been out there regarding MALDI-TOF and identification. Okay, so what difference does this make, and why is this such a big deal in the field of clinical microbiology? So this is not, for the most part, changing the initial specimen and requirement to grow it on a plate. But what it is doing is dramatically reducing the amount of time it takes for an identification to be produced by the lab. So in contrast to traditional methods that are biochemically based that require at least overnight growth, the MALDI-TOF produces results in mere minutes compared to hours or days. And so, although this is not actually real, I would encourage the MALDI manufacturers to consider including a black box warning on their product, because initial use of it can be associated with symptoms of hyperventilation hysteria amongst lab personnel, including lab directors. Okay, so just to summarize the platforms that are available, what I'm showing you right here is the Bruker MALDI biotyper. That's the first instrument that came to market. That's actually the instrument that we use here. And so they have flavors of their databases. They have an FDA-approved database that covers gram-negative organisms, and then they have a separate research use only database that has, as you can see, over 2,000 different species present. They also sell separate Aureus, mycobacteria, and fungal databases, and we'll get into that a little bit later. In terms of how it makes its ID, the Bruker biotyper basically matches spectra. It looks for things that are in common and things that are different, and it produces a score value. And so these thresholds are important. A score value of greater than or equal to 2 is a definitive identification to the species level, and a score greater than or equal to 1.7 but less than 2 is considered identified to the genus level. I'm going to also talk about the second platform in my talk, so I just want to introduce it so you can understand why it's a little bit different. The first thing, it's a little bit different in terms of size. The Bruker lives on a bench top. The uwvideo basically looks like a Coke machine. And so the poor guy who came to install it is really tired of people cracking jokes about putting in money and getting out Coke machines. So it's literally that big. So the bioMérieux, both systems got FDA clearance in 2013. So the IVD FDA-cleared database includes both gram-positive, gram-negative organisms, yeast, and anaerobes, and it also has a research database, but their research database includes mycobacteria and filamentous fungi. And so in contrast to the Bruker platform, which uses spectral matching, the bioMérieux platform uses what they refer to as advanced spectral classifier, an advanced spectral classifier. And basically, all the spectra then are generated or translated into numbers and sorted into bins. And the different bins are weighted based on sensitivity and specificity for calling certain organisms. So instead of getting a score value, you get a % probability or a confidence value. So obviously, the fact that it's fast is great, but if it's not accurate, then who cares? And so there are many, many papers that have been published looking at this, but this is a table that was published by Robin Patel in 2013. And bottom line is, without getting into the details, is that MALDI-TOF overall displays equivalent or if not superior performance compared with traditional identification methods, but does it in a much faster manner. So I mentioned that we evaluated this maybe I didn't, quite a long time ago, and one of the technologists who was involved in the initial evaluation, one was Susan Turner, who is our lead in bacteriology, and the other was Bruce Olness. And so he used to refer to this as if you had a bug and you didn't know what it is, what would God say? What does God say it is? So there's this, I think, perception amongst lab staff that the MALDI is somehow infallible. And so I want to present a case of what happened here to show you how it is not indeed infallible. Okay, so we had a 62-year-old male who previously had been on holidays in Thailand. He suffered a heart attack when he was there, which is a total bummer because you're in Thailand and you've only been there for 3 days and you're hiking around waterfalls and stuff, and all of a sudden you have a heart attack, which might be kind of what led to the heart attack. And he was hospitalized there for a week. 1 month after he came back to Seattle, he developed symptoms of a urinary tract infection, and his primary care physician prescribed him nitrofurantoin. There was no improvement in his symptoms after 4 days of taking the antibiotic, and so he went back to his doctor, and a urine specimen was obtained for culture and sent to our lab here at UWMC. Okay, so after 24 hours, pinpoint growth was noted on blood agar. So that's the sort of common situation that we would see for things that are gram positive. We're not seeing growth on our gram-negative media. It's sort of pinpoint looking. But then after 48 hours, we saw that there was pure growth of what clearly now was the gram-negative that had grown up sufficiently. And it was growing on both blood agar and McConkey agar, which are standard microbiological media. It was oxidase positive, and the MALDI kicked out an ID of Burkholderia thailandensis. And it gave a score value that, although wasn't sufficiently high to be identified to the species level, would be identified to the genus level. However, it's important to point out that many clinical labs that are using these systems have validated lower score thresholds. So depending on the organism, some labs will actually accept a score value of greater than or equal to 1.8 as a species level identification. So the problem is that this was not what it really was. And so at the time, there's a joke in the lab that when I leave the country is when really bad stuff happens, and there's an N of 3 to prove this. But in this case, Tanis Dingle, a former fellow, was doing bench rounds because I was out of the country, and the techs said, hey, you check this out, this is so cool, we have a Burkholderia thailandensis on a urine culture. She said, close the plates and bag them up, and thanks to Tanis's astuteness, she rightly guessed that we shouldn't believe the ID, and this was in fact Burkholderia pseudomallii, which is a select agent. It's the causative agent of melloidosis. And so depending on what definition you use of exposed personnel, and our public health folks included the FedEx guy who walked through the lab with a box, 21 employees were exposed, both high risk and low risk. Certain employees were required to do things that maybe didn't make the most sense, like come in on their holiday days off to give blood cultures and all that stuff. So it was a big mess. And so the bottom line is that these instruments are not infallible. So in addition to this, there are other problematic organisms. So some really commonly encountered misidentification issues or known identification issues are that MALDI will overcall strep pneumo. So viridin strep, of which there are many, it'll tend to call them strep pneumo. It can misidentify Shigella as E. coli, and vice versa, depending on what database you're using. And also misidentification of saprophytic Neisseria species as Neisseria meningitidis. Obviously, select agents are an issue. In the standard Bruker database which we use, there are no select agents at all. So there's no Burkholderia pseudomallii, there's no Brucella, B. anthracis, any of that. And so these are huge gaps. You don't have a database that you just get out of the box that's going to be able to help you identify these bugs. Some other issues are that it's not reliable for serotyping Salmonella, and it also has difficulty resolving members of different complexes or highly closely related groups, for example, the Bacillus cereus group or B. specia complex. Okay, so I wanted to give you, so that's, I'm really, I guess one way to think of my talk is it's kind of like the new micro board exam. It's like a little bit of everything, but not in great detail. So I'm just going to really talk about how MALDI's changed our practice, and I want to give you one example of how it has changed our workflow and allowed us to dramatically improve our turnaround times. So Stool culture, for anyone who's ever been in a micro lab, is an involved process. We look for a variety of stool pathogens. We have to look for these using a variety of different media that are both selective and differential. And so previously we had been using pretty standard media, enteric media used for the recovery of Salmonella and Shigella species, so hectone, enteric, and McConkey augurs. But the problem with these is that there's lots and lots of normal flora that can mimic pathogens on these media, so for example, Proteus or Citrobacter. And so what happens in labs is that you basically have all these mimics that you have to rule out. So you have to set up additional tests that require additional growth in vitro, and it's just, there's lots and lots of work associated with these cultures. So the question was, could we combine MALDI-TOF with a better media for the recovery of Salmonella and Shigella species, and how would that impact what we do? So a technologist here in the lab, Katie Schwartz, did a study, which she presented at ASM, where she took 2 stool specimens and compared the performance on a chromogenic media that we chose to study, the hardy chrome, Salmonella Shigella agar, compared with our traditional workflow. And so rather than subculturing the organisms that grow on the chromogenic media and identifying those, we did perform MALDI straight off of the chromogenic agar. And we didn't know if that would work or not work, because chromogenic agar, basically the colonies take on a color, just like they do on the hectone agar. And she compared this with our routine workflow. And so obviously if we got an identification of E. coli, as I've mentioned, MALDI isn't very good for E. coli versus Shigella, we would have to do traditional ID at that point. But we cut out, if this were to work, we would be able to cut out all these tubes that we have to do for all of these suspicious colonies. And so what we found was, by using this agar, it actually had improved performance relative to our traditional method. We recovered more Salmonella and Shigella isolates. And what we found also was that we had improved specificity, meaning that we had less normal flora that we even had to go to the trouble of doing a MALDI on. And the key of this was that basically a negative culture could be resulted 2 days earlier than with our traditional methodology. The turnaround time for reporting a Salmonella, which is a reportable organism, was reduced by a day. And we found almost 50% cost reduction associated with using this new approach. Okay, so I've showed you hopefully how we can harness MALDI to improve what we're doing, but the bottom line is, how much does this instrument cost? And so the instruments themselves are expensive. They're in the order of $200,000 to $250,000. Now, for people that are used to using plates and platinum loops that we flame because we don't want to pay for disposable loops, that's a lot of money. So is this something that's really feasible for labs to bring in? And so the bottom line is that MALDI-TOF is cost effective for routine organism identification. So in this study published by Karen Carroll's group at Johns Hopkins, they found that they had an annual cost savings of around $100,000 per year. And in our data, what we found was that we had annual savings of $94,000 per year. And we reduced the number of isolates that we had to send for sequencing. Now, I want to point out that although we had reduced labor costs, this ability to reduce the amount of work involved, it's just basically silly work, overworking, allowed us to take on additional testing without having to add FTE, and it allowed us to also bring on new tests without having to add FTE. Okay, so up to now, I've just been telling you about the pretty routine stuff that we're doing. And so anyone who has small children will know who this lady is over here. But this was my Bible, because it was all about establishing the routine for small children. But we're going to break the routine now for the rest of the talk. And we're going to talk about 3 different things. We're going to talk about MALDI as it pertains to direct blood culture identification. MALDI and its ability to identify Mycobacterium species. And lastly, I'm going to tell you about some very preliminary work that we've been doing to use MALDI to potentially detect Vancomycin intermediate Staph aureus. So I'm going to begin with blood culture identification. So why are blood cultures so important? Well, they're so important because they're what we use to diagnose the etiological agent of bloodstream infections. And so obviously bacteremia is the presence of bacteria in your bloodstream. But what makes the news is sepsis, which basically have a dysregulated inflammatory response to infection. And so sepsis is associated with very high mortality rates. And unfortunately, sepsis due to nosocomial pathogens or infections that are acquired in the hospital are associated with higher mortality than those that are acquired in the community. The number of patients that are being hospitalized in this country with sepsis is increasing. And for patients who are septic, they have 75% longer hospital stays than those with other conditions. So in 2008, it's estimated that there was $14.6 billion being spent on hospitalizing patients with septicemia. And so obviously, that's unlikely to go down in terms of the number at its current rate. Okay, so one thing that's key, though, is the impact of delayed therapy. So this is a paper that's widely cited that's looking at a specific issue, and this was a large retrospective cohort study done across 10 hospitals, 14 different ICUs between 1989 and 2004. And so bottom line is that for each hour that appropriate antimicrobial therapy is delayed, your mortality increases by 7.6%. All right, so think about that. That's really staggering, especially in the context of what I showed you before, where we're rocking it like microbiologists. And so just to show you how what microbiology does is not really consistent with the clinical needs of managing sepsis, we do blood culture, which is great because we get the bugs and we can test them, but we have to wait for them to grow up. And then when they've grown up, we do a gram stain, and then we have to wait in the standard algorithm. You have to wait for it to grow on the plate. And then if you're not using MALDI, you would have to also wait longer to get a definitive identification. Now, there are some tricks that can be done here phenotypically, low cost, to try and get a faster turnaround time. There are also, importantly, molecular tests that are available now that can identify organisms directly from blood culture, but I'm not going to talk about those today. Instead, I'm going to focus on MALDI and discuss whether it has the ability to also do direct identification from positive blood cultures. Okay, so the answer is it can, in that Bruker, the manufacturer of our MALDI-TOF instrument, has a blood culture specific module. And so the difference between this and the regular module is that it looks over a slightly different mass range. Instead of looking from 2 to 20,000 Daltons, it looks between 4,000 and 20,000, and also has a lower threshold for being considered identified to the species level. And Bruker also sells a kit known as the Sepsi-Typer kit, which basically has washing and lysis steps and takes about 5 minutes to perform the processing, and then you do a standard extraction, which is just ethanol, formic acid, acetonitrile. So bottom line, 20 minutes or so processing time. And so Raquel Martinez, a former fellow of ours, and Elizabeth Bowerly, a master's student who now works in the lab as a MLS, embarked on looking at the ability of MALDI to directly identify bacteria from positive blood cultures. Now, this was done as part of a larger study with Dr. Fong, Harborview, looking at molecular methodologies too, but I'm just going to focus on the part that has to do with MALDI. So Raquel and Elizabeth looked at 159 positive aerobic blood cultures. So we didn't include anaerobic blood cultures in this study because of the study design and other assays we were looking at. And so what we found is pretty standard with what most labs see in terms of our organism distribution. Staphylococci are what we see most commonly, followed by E. coli. And so what they did was compare the identification that they got directly from the blood culture to that which they got from the bacteria when they were growing in colonies. And so just to highlight the performance for broken down by gram positives and gram negatives, what you can see here is that if you look to the right where you have the correct identification, and these are all to the species level, it does really well for Staphylococci. We saw for coag negative staph 84% correctly identified, which is actually higher than was published in other studies with this method, possibly because we did some additional washing steps. And it seemed to do very well with the exception of, as we would expect, viridin strep. So the numbers don't add up because I'm not showing you all the organisms. I'm just showing you the ones where there were issues. So the viridin strep affected the performance for streptococci, but the non-viridin strep, so the beta hemolytic strep, worked. And so for the others, so the micrococcus and some other gram-positive organisms, micrococcus didn't seem to work that well. And so I would say that we don't feel terribly confident about how well it performs for these types of organisms. But bottom line is it does very well for staph and very well for enterococci, which are very commonly encountered in clinical labs. In terms of its performance for gram-negatives, it did excellently for Enterobacteriaceae, so enteric organisms again. I'm just showing E. coli, but it really performed excellently for Enterobacter, Citrobacter, other common organisms. Unfortunately, it didn't seem to do quite so well for the non-Enterobacteriaceae. And these are the ones that we'd really like to be able to identify quickly because, for example, Stenotrophomonas multifila is intrinsically resistant to carbapenems, and it's not unusual for patients that come in to be put on carbapenems as part of empiric therapy. And so we'd want to be able to catch those and let the physicians know that that's what's causing their infection. So it did not perform as well for non-Enterobacteriaceae. Okay, so what's the upshot of using MALDI directly to identify blood cultures? So obviously the pros we found in our study, and this has been consistent in other studies, that when you do get a species identification, with the exception of viridans strep, it's pretty accurate. So we didn't have any other misidentifications in our study other than with the viridans strep. If it didn't work, it just didn't work. It didn't call it the wrong bug. And so overall, we found in our study, taking the gram-positives, gram-negatives, and yeast, which I didn't talk about together, we found that 80% of organisms were correctly identified to the species level. Now, just to put that into perspective, that's an organism ID within a half an hour to an hour, compared to waiting, at least 24 hours, potentially longer to have a definitive ID. It's very cheap to perform, right? The reagents are really low. There's fairly minimal tech time involved compared to, say, some of the molecular methods that are available. But the problem is that it struggles with polymicrobial cultures. So meaning, if there's more than one bacterial species present, it struggles to identify them. And so 8.2% of our cultures in our study were polymicrobial, and although the MALDI was able to identify one of the bugs that was present in around 76% of the time, it's not able to identify the second organism. So it doesn't do well for polymicrobial cultures. And this is really key. It doesn't provide any information on antimicrobial resistance or susceptibility. So even if we tell you it's a Staph aureus with this method, we can't tell you whether it's MRSA or MSSA, which has important ramifications for therapy. However, I think that it can be used as part of an algorithm that includes molecular testing, that maybe this could be something to reflex to. And I know in some very large medical centers that, 8-center systems, it becomes prohibitively expensive to do molecular methodologies to assess this, so they're often using MALDI. Okay, so that's great and all, but does any of this make a difference? So one way to think about it is you might have a preference about what method you'd like to do, but is it really all just the same thing? So the answer is it does make a difference, but it only makes a difference with the intervention of antimicrobial stewardship. And so we're very lucky here. We have a great antimicrobial stewardship team here at the U. Rupali Jain is the ID pharmacist. Paul Pottinger is the infectious disease medical director. And so antimicrobial stewardship is key. If there's multiple studies that show that if you do rapid identification of positive blood cultures, it does nothing unless you have stewardship involved. If you just put the result out there, you can't expect that anyone's going to act on it. And so the bottom line is it does make a difference. And so these are some studies that have been published with regards to using MALDI-TOF performed directly on positive blood cultures. So the first study was by Sarah Cosgrove's group at Hopkins, and this was a theoretical study. And so looking at their retrospectively, they calculated that they'd be able to avoid 58 days of therapy per 1,000 patient days, and that there would be around 45% or so of patients would have improved time to appropriate therapy. But that's theoretical. What's actually been shown? So there's a really nice study that was performed by Jim Musser's group, and it showed that using direct identification of organisms in combination with antibiogram data, and this was performed using MALDI, obviously, that they found a reduction in all-cause 30-day mortality from 21% down to 9%, around $20,000 or so dollars saved per patient, which if you multiply it out in their large system would result in a cost savings of $2.4 million, which are really huge numbers. But just to show you that it's not just necessarily all having to do it at the direct identification level. If you partner with stewardship closely, which Dwayne Newton's group did at Michigan, they found that even when they just did MALDI off of the colonies that were growing, they didn't process the bottles or do any of what I just described, they just did their identification off the colonies, but made sure that they engaged stewardship and and they were involved in changing therapy. They observed, again, pretty big reductions in optimal antibiotic therapy, decreased mortality, although not to the same extent as that was observed here, possibly because of the difference between, most likely due to the difference of doing it directly versus waiting for the colonies to grow, and a decreased length of intensive time in the ICU. So really, it does make a difference. Okay, so switching gears, I'm going to talk about now going from a specimen, a positive blood culture, back to identifying an organism. And so I'm going to tell you about mycobacteria. Now, obviously, as Dr. Schertz mentioned, I did a postdoc working on TB, so mycobacteria have a special place in my heart. But why do we need to identify these organisms? So the Mycobacterium genus actually has over 150 different species. And although there are some really great molecular detection strategies for this, designed really for tuberculosis, they're not perfect. And so the bottom line is we still need to perform culture for mycobacteria. We can't just rely on molecular detection of these organisms. And so the reason you have to identify these is that there are important treatment differences between what we refer to as non-tuberculous mycobacteria and TB. And so, for example, there's differences between infection treatment for infections due to Mycobacterium fortuitum versus M. cheloni. As a result, the American Thoracic Society and the Infectious Disease Society of America recommends that all clinically significant mycobacterial isolates be identified to the species level wherever possible. Okay, so how do labs typically identify Mycobacterium species? Well, traditionally, labs would have performed phenotypic testing. These are based on growth characteristics, biochemicals, pigment production. And when you think about mycobacteria, there are rapid growers that can grow within a week of being subcultured, and then there are slow growers that can take weeks and weeks to grow. So trying to do phenotypic testing on something that takes weeks to grow is just not really ideal. HPLC has also been used. It's not widely available, but by all accounts, labs that have it find it to be quite good. And then there's molecular assays. Now, there are probes that can be done on positive cultures, and those don't take days. Those are rapid. They're great, but what you can see is that they only cover these species, these 5 species here. Although these are actually complexes technically. So that leaves us with having to do sequencing, which is fantastic. It's really accurate, but bottom line is it still takes days, and it's very expensive. And so as much as our clinical colleagues might like to think that these are the types of folks that work in the lab, we have real people working in real labs, and we don't want to stick them with having to do a ton of manual labor for no gain, or if it's inaccurate. So bottom line is that I think there's a real need to have rapid, cheap identification of mycobacteria. So bottom line is if you could use MALDI, you could do this very cheaply because MALDI is so quick, you could do it very rapidly. And we know from MALDI in other organisms, it's very specific and has high accuracy. But with regards to mycobacteria, there are some important considerations. First is if you have a slowly growing mycobacterium species, you don't know if it's a TB or not. And tuberculosis is it's an important cause of laboratory-acquired infection. So the infectious dose for TB is very, very low. If it's not handled appropriately, you can become infected. The cell wall of mycobacteria are different than the cell wall of many other pathogens. They have very lipid-rich cell walls, so that's also a factor. How well would it work on a MALDI when you have this really big, thick cell wall around the bug? How reproducible is the method? How much organism do you actually need when you've got this big, thick cell wall around? And then what's the story with databases? Because, and I'll get into this a little bit later with regards to what's available from the manufacturers. So bottom line is, when we looked at this, and I was really, really lucky to have just a phenomenal resident, Cheryl Mather, work on this project. This was her baby, and she just did a fantastic job. And when Cheryl started looking at this, what we found was that there was really very little published on the performance of MALDI to identify mycobacteria. And so this is really it. When she started, these are really the only publications that were there. So I'll draw your attention to 2 salient features. The first is that the number of strains in the databases were pretty small, like less than 50 in most cases. And the second thing was just the diversity of different extraction methods were being used. Now, somebody back in 2006 decided it was a good idea to put mycobacteria directly onto a plate and just stick on your matrix and off you go. That's not a good idea because matrix doesn't actually kill Mycobacterium tuberculosis. And although actually they managed to get it to work, that's not something that we want to do. Similarly, you can have sort of hit or miss inactivation in ethanol, so that's not necessarily an approach that we want to do. And so when Cheryl looked at the literature, what she saw was just this smorgasbord of methods, including things like micropestals and all sorts of stuff. And so what were the manufacturers saying? Well, the manufacturer of the instrument that we use had a procedure, which is great. They have a procedure, they have a database, let's just use that. But when you actually look at what their recommended procedure was, it involved you to do extensive manipulation of the culture before you ever inactivated it. And that's just not something I think that is safe or practical for labs to do. You don't want to be spinning and washing a bunch of times before you actually kill off your TB. So what we decided to do was to build our own database. And so what Cheryl did was, rather than trying to build a database that covers all 150 different organisms, instead she elected to choose organisms that were specifically highlighted by the American Thoracic Society IDSA guidelines as being most clinically significant. Obviously, those guidelines were about non-tuberculous mycobacteria. You have to include TB. And so we also included some other organisms, particularly M. lentiflavum, that we commonly encounter in the lab. Now, everything with a line through it are bugs that we didn't have access to, and I just want to take this opportunity to thank the molecular section of the lab, because all of these isolates that Cheryl was able to use had been definitively identified using DNA sequencing, and were in the stocks in the molecular section. Okay, so what Cheryl did was she basically developed her own extraction method, and so it's shown here on the right. And so the first step here is to basically inactivate the organism, which is really what any good method should be doing. Just kill the TB before you start monkeying around with it. And so what she did was to transfer only one loopful of mycobacterial biomass. Now, importantly, some other procedures had used up to 10 microliter loopfuls, so it was quite a small amount, in deionized water with silica beads. And then she heated those for 30 minutes and then added ethanol, did a bead beating procedure, and then did a standard ethanol formic acid acetonitrile extraction. So compared to other published methods, and particularly compared to the manufacturer recommended method, this was remarkably simple. Cheryl then used this method to build a database, which I've shown here. And so you don't have to pay attention too much to everything that's in here, but the bottom line is that she was able to build a database containing 123 different strains across the species that she targeted. In some cases, we didn't have very many of them. In some cases, we were able to dramatically increase the numbers. And so what she chose to do was to combine our database with the existing manufacturer database and see if it works. But there's no point in doing all of that unless you know that your extraction method is going to inactivate the organisms. And particularly, this is really focused on TB. And so what Cheryl did was, after her inactivation step, she inoculated both solid media as well as mycobacterial liquid media with 100 microliters of sample, and incubated her plates and her broths for 6 weeks, I believe, or 8 weeks. I can't remember which one it was, the appropriate time for recovering mycobacteria. And so she tested 5 strains of TB and the other organisms shown here. And what she found was that there was no growth. So this is great because we have a method that's effectively inactivating our organisms. And so then the important thing to do is to say, does it actually work? So it's all well and good having this simplified extraction method, but if it's not going to work, then what's the point? And then at the same time, we happen to have an instrument from the other manufacturer, the other commercially available manufacturer, the uwvideo in the lab. And so Cheryl decided it would be interesting to compare the performance of her database with that of the other system. Now, importantly, that other system actually also includes a relatively simple, simplified extraction procedure as well that has some differences with ours, but she wanted to see how they stacked up. So the bottom line is that both our Cheryl's UW procedure and the Butler-Wu extraction procedure produce spectra of similar quality. So down here on the x-axis are the different parameters that are commonly used to assess spectral quality. And what you can really see is that between the UW protocol and the Butler-Wu protocol was that these were fairly equivalent. The only place we noticed a difference was in terms of the maximum intensity of when you ran both extracts from both protocols on the VITEC-MS instrument. And we don't really know what the significance of that is. But bottom line is that the UW extraction procedure was generating spectra of high quality equivalent to that of a manufacturer recommended protocol. Okay, so how did this stack up? So Cheryl ran many isolates, and this is a busy slide, so I'm just going to highlight the pertinent features in red. And bottom line is that when Cheryl tested her database, and so she combined her database that she built with the manufacturer database and ran her isolates using her extraction method, she observed that almost 95% of strains were identified correctly to the species level. The other manufacturer's system and extraction performed very, very similarly. Cheryl also took her extractions and ran them just against the Bruker recommended Bruker database, and what you can see is that the identification numbers were, there was a poor identifications obtained. However, interestingly, regardless of what database you used or what extraction method you used, we observed 100% correct identification of tuberculosis. And so there's just to highlight one last pertinent feature. We were correctly able to identify chelone from the M. abscessus group for both databases and both protocols. Okay, so lastly, I'm going to talk about something completely different. And this is, as I said, very preliminary work. And this is, we are trying to see if we can use MALDI-TOF to detect Vancomycin intermediate Staph aureus. And again, I have Cheryl, who's expressed interest in working on this, and this is really her baby, and she's running with this, and we've been really lucky to partner with Brian Worth, who's a fabulous ID pharmacist in the pharmacy department here, and Shobini, who's an ID fellow from Australia. So there's a nice little group of us working on this. So vancomycin, and why do we care about it? So I'm sure everyone here has heard of vancomycin. So vancomycin is really the mainstay in terms of treating serious infections due to MRSA. It's a glycopeptide antibiotic that inhibits cell wall synthesis, and this is an antibiotic that's active against gram-positive organisms. And so in terms of what breakpoints we use to call something susceptible, the Clinical Laboratory Standards Institute has a breakpoint set of susceptible bugs or isolates that have MICs less than or equal to 2 micrograms per mil. And this number, less than or equal to 2 is important, and it'll come into play as I continue through the remainder of my talk. It sets intermediate criteria as MICs of 4 to 8 and resistance criteria of greater than or equal to 60. So the reason the 2 is important is that it has implications with regards to the pharmacodynamics of this drug. So the optimal pharmacodynamic parameter that has been shown to correlate with treatment success for vancomycin is the area under the curve over the MIC, and that ratio has to be greater than 400. So I know that people aren't really versed in this, and the MDs in the audience at some point in their med school learned about this, but I know it's not something that you guys think about a lot. But the bottom line is that you just have to remember that this number greater than 400 is what seems to correlate with success. The problem is that when they modeled this, the probability of target attainment, so the probability that you're going to have an AUC over MIC ratio of greater than 400 is very, very variable when your MIC is 2. So remember, when your MIC is 2, you're still considered susceptible. And so your probability of target attainment can range from anywhere from 0 to only 57%. And that 57% is at very high doses of vancomycin, and if you administer those doses of vancomycin, up to 35% of patients getting those doses will have nephrotoxicity. So, and this is even when the vancomycin trough is looking peachy like it's supposed to. And so to show how MIC correlates with vancomycin failure or success, there's this very nice meta-analysis that was published not too long ago, comparing the results of all these studies, and they stratified, they looked at a number of parameters, but I'm going to focus on vancomycin failure or success. They stratified it based on the MIC that was obtained. And so what you can see is that for patients who had MICs that they characterized as high, which is greater than or equal to 1.5. And so 1.5 usually in a lab would be rounded up to 2, because for MIC testing you usually report to the next doubling dilution. And they compared those to patients who had MICs that they considered to be low, which are MICs less than or equal to 1. And what they found was that patients with high MICs, the odds ratio was 2 for vancomycin failure. Okay, so on top of that, you can then layer this issue with vancomycin intermediate Staph aureus. So I told you that there's susceptibility criteria for vancomycin intermediate Staph aureus of 4 or 8. I'm not going to talk about vancomycin-resistant Staph aureus, or VRSA. These are bugs that are very, very rare. There's only been 3 kind of clusters of infection, and they are due to Staph aureus that has acquired a VAN-A gene from Enterococcus faecalis. So I'm not talking about that. Instead, I'm talking about vancomycin intermediate Staph aureus. So what's really interesting about these strains is that they look different than regular Staph aureus. So they have this thickened cell wall. They can also have decreased autolytic activity and other features. And so the problem is that in terms of visa, it's not just due to a single change at the genetic or transcriptional level. So for MRSA, we can just detect MEK-A. For certain gram-negatives, we can detect the presence of certain carbapenemases. We can't do that for vancomycin intermediate Staph aureus. But they do, many of them, most of them in fact, have this thickened cell wall. And what's that's believed to do is to basically reduce access of the antibiotic to the target. So in particular, at the tips of the septum that's forming, there's reduced ability of the drug to reach that target because of the thickened cell wall. So that's vancomycin intermediate Staph aureus. So you should think, okay, well, there's CLSI criteria. If it's intermediate, that's great. We just report it and problem solved. Well, the problem is this problem with H visa. So H is for heterogeneous. And so the issue with H visa is that the resistant population is present at a frequency of 10 to the minus 5th to 10 to the minus 6th, sometimes even less frequently than that. But when we do susceptibility testing in the lab, we use an inoculum size of 5 times 10 to the 4th. So what that means is you could potentially be missing this subpopulation of resistance when you're doing your susceptibility testing. And so there's really only one standard method that has been proposed and proven as the gold standard for detecting H visa, and that is this population analysis profile. And as the name suggests, it is one of these. It is time consuming, it's labor intensive, it's not something that labs can actually really be doing in the clinical setting. And essentially what they're looking at is the population analysis profile over the area under the curve compared to a control strain. And if it has a ratio of greater than or equal to 0.9, it's considered to meet the criteria for being H visa. Brian in his lab does this, and it's a lot of work. Okay, so far I've told you that really, why would we even care about H visa? Because we already know that if you have a vancomycin MIC of 2, that you're more likely to fail vancomycin therapy. Well, perhaps it's not just a numbers game after all. So this is a retrospective multi-center study that was published looking at patients between 2004 and 2012. And they had 122 patients with MRSA bloodstream infection. And they had 61 matched pairs. And so they did PAP analysis on all of these retrospectively and looked at the outcomes and compared the outcomes in people that were confirmed to have H visa and those that are confirmed to have true vancomycin susceptible Staph aureus, so VSSA. And so bottom line is for every parameter they looked, the patients that had H visa did worse. So in contrast to the twofold odds ratio for vancomycin failure that you saw if the MIC was 2, if H visa was present, those patients had 11 times greater odds of failing vancomycin therapy. They had an increased length of stay of 8 days. So remember, it's about $1,500 to $2,000 for each day you're in the hospital. Multiply that by 8, that adds up. And I think this is really key. In this study, they didn't observe any relationship between the AUC over MIC and vancomycin failure. And so when they looked at patients that had an AUC over MIC of less than 400, they had the same rates of vancomycin failure as those patients that had AUCs over MICs, AUC over MIC of greater than or equal to 400. So that would argue that perhaps it's not the MIC that matters so much, it's actually the presence of H visa. So what do we know about H visa in terms of its prevalence? It varies widely between studies, and part of that is how these studies are performed. Some of these studies, they freeze the isolates and they don't try and enrich for these when they're pulling out the bugs, and then they make the conclusion that there's no H visa here. Some studies have claimed that they have prevalence rates of up to 50%. But when, as we know, unfortunately, when H visa strains are tested using standard susceptibility methods, they can have low MICs. So we can't just use a higher MIC as a criterion for looking for these strains. So there's a real need to have a way to identify visa and H visa. And there are lots of different culture-based methods that have been published, but they have very variable performance and can require up to 48 hours of culture incubation. Similarly, the Infectious Disease Society Guidelines of America state that they don't endorse changing therapy if the MIC is 2, but on the other hand, they say if you detected visa that you should change therapy. So what was the goal here? And I'll just have a few slides showing you some pilot data on this. The goal is to develop a rapid MALDI-TOF-based tool to detect visa or H visa from bloodstream MRSA isolates. And we believe that if we could do this, and we could do it through MALDI in a rapid manner and be able to report that, we would be very interested to see if those patients have a more positive outcome. So Cheryl did a little pilot study a while back where she took 13 known visa strains and 12 presumed VSSA isolates, we hadn't confirmed them at this point, and compared the spectra on blood agar compared to Mueller-Hinton with vancomycin. And the reason she chose Mueller-Hinton was because Mueller-Hinton is something that most labs have. And so what she observed was despite looking at many, many peaks, there was really only, there was no unique peak that was observed to be different in terms of presence or absence between VSSA and visa on blood agar. But on Mueller-Hinton with 0.5 micrograms per mil of vancomycin, she observed one peak that was present at a little over 6,000 Daltons. But the problem was that the peak was absent from 25% of the visas that were tested. So it looks like it could have something to do with it, but it's not going to be all based on this one peak. But when she was looking at these spectra, she saw that there was differences with respect to the intensity of the peaks between VSSA and visa. So very, very recently, and these are really hot off the press data, so we have looked at 16 confirmed visas and 47 confirmed VSSAs, and these were all confirmed in Brian Worth's lab by the gold standard method. And so Cheryl basically tried to cast a very, very broad net. And so her criteria were very, not too stringent to begin with. She basically observed that there were 394 peaks that were present in at least one of the spectra that she obtained for each isolate on both pieces of media. And so then she tried to narrow that down. And so ultimately she found that there were 168 peaks that were shared between each media type within a class, so within visa or within VSSA. And so when she looked at these and she picked a threshold of having a intensity that's 1.3-fold greater, what she observed was between visa and VSSA that there were peaks that changed in intensity between these. But for VSSA and H visa, there weren't any peaks that differed in intensity. And so that suggests that that really just speaks to the issues with trying to detect H visa. But regardless, she built a training set, and she picked 10 visa isolates and 10 VSSA isolates, and she chose these isolates based on MIC and their differences in their MIC to PAP value. And I'm not going to go into details too much because I'm short on time. She picked criteria that were consistent with what had been shown in literature for other examples of trying to use peak presence or absence or intensity. And so she tested the remainder of the visa and VSSA isolates. And what she found was she saw 100% sensitivity for visa detection and 84% specificity. So meaning that it called visa correct the whole time and was mostly right about calling VSSAs. And interestingly, when she ran 47% the H visa strains, even though they weren't in the testing algorithm, the training algorithm, it actually called 47% of these visa, even though she didn't really, when she looked at peaks, like I showed you in the previous slide, there wasn't a huge difference. For the blood agar, it didn't work quite as well, but interestingly, even with blood agar, she was able to observe that 27% of H visa strains were called as visa. So in conclusion, I hope I've convinced you today that MALDI has revolutionized clinical microbiology. It's allowing us to change how we do routine identification. It's allowing us to look for things we never thought we could look for before, like visa and H visa. It's made dramatic impacts in terms of our ability to identify organisms that we routinely see. It has the ability to reduce the time to identification of blood culture isolates, accurately and rapidly identify mycobacteria. And just really quickly, my acknowledgements. It would be remiss of me not to point out the immense contributions of Dr. Cookson and Hoofnagel, without whom we would not have had MALDI here. They were instrumental in getting the MALDI instrument here for the initial eval. I think I've mentioned most of these folks. And last but not least, everybody who's involved in the UWMC micro lab. And with that, I will take, I think I have a few minutes left for questions. Jenny.
Excellent talk. My question is, what's the potential for high volume rapid throughput on this? And I ask because one of the challenges we have with platelet therapy is that we're primed to culture apheresis platelets, but they need to be 24 hours old. Typically, a blood center will hold on to them for a minimum of 12 hours, and in some cases 24 hours, before they issue them to hospitals, and then you run into problems of out of date.
So the question is, it's a great question, and I'd just like to say that I'm not here to answer questions. What was it that Marshawn Lynch said? I totally screwed it up. I was going to say, I'm just here so I don't get fined. I totally screwed it up. That's a great question. So the question is, what's the ability to do high throughput on this, because there may be a need in blood banking to turn around, to release units faster. And so bottom line is there's great potential for high throughput. The system that we use, you can run up to 96 organisms at a time. And so on other platforms, they have the capacity to actually run even more at that time. So there's very high throughput. And it's very short assay time.
So how many samples could you do an hour, do you think?
Oh, gosh. I don't think we've looked at that, but there's data that I've seen, and I want to say it's a couple hundred, but the question, sorry, was how many could run an hour. I believe it's up to a couple hundred, but it all depends. If you have more instruments, you can run more. So some big labs have like 2, 3 instruments, 4 instruments. Okay. Noah?
Great talk, thanks. So one of the reasons 16S is such a useful target for identification is because we have so many sequences, and there we have community resources for reference data. Are there any prospects for a public database of mass spec spectra, or is it just going to be sort of a bunch of closed platforms that vendors provide or the individual labs develop for themselves?
So that's a great question. The question is, what's the potential for having open source platforms, open source databases for microbial ID by MALDI, or are we just sort of limited to what the manufacturer gives us or what we all develop? And I would say that, so Anna Lau, who's at the NIH, has developed, the NIH themselves have developed a number of databases, including for fungi and mycobacteria, and those are publicly available. There is no reason that you can't share the database that you build with anybody else if you have the ORUO platforms. So there is potential. I just don't think there's a whole lot of that happening to date, but there are NIH, because taxpayer money built them, they're freely available to whoever wants them.
So, great talk. So you have one machine, you have a lot of methods and thousands of species. So if the FDA got their way and regulated lab-developed tests, how would you even begin to comply with such a versatile and sort of wide-ranging platform?
So the question is, how would one comply with proposed new regulations proposed by the FDA, where this would be considered an LDT? Because this is recorded, I'd like to plead the fifth. Is that the terminology used here? That's what I'm going to do. But I'd be happy to talk to you about it afterwards. If there's not any more questions, then thank you guys so much for your time.